I'm Jonah Comstock, editor-in-chief of pharmaphorum. This is my last meeting of the day on day one of JP Morgan in San Francisco. I'm at the Axiom Hotel, and I'm joined by Albert Seymour. I'm joined by Albert, it's the last meeting of the day. Joined by Albert Seymour, the CEO of Seamless Therapeutics.
Yeah, nice to meet you. Thanks.
Great.
Happy to be here.
Yeah, thanks for meeting with me. So tell me a little bit about Seamless and what's the elevator pitch?
So Seamless Therapeutics is built on the foundation of using an enzyme called recombinase. And these are enzymes that have been used in the field since the 1980s. And what's unique about them is that they can make an edit into a cell that is extremely precise. Mm-hmm. And what I mean by that is that it can make an edit so that it's right to the nucleotide.
Hence Seamless.
Hence Seamless. That's the name of the company. The challenge with that for a long time was that they recognized a very specific site that they would bind to. That site does not exist in the human genome. So you could never really think about them as a therapeutic. Mm-hmm. But technology coming out of Dresden University in Dresden, Germany, Frank Buchholz's lab and Felix Lansing, our CTO, what they figured out how to take and retain all the properties of the enzyme, but program it to bind where you want it in the human genome. Thus now making these available and amenable to therapeutic intervention. And that's what Seamless is now developing, utilizing these enzymes. We're the only company in the world that has that ability to program them to go where we want it to go in the genome.
So that sounds like something of a platform play. What do you plug into that? What are some of the first kind of applications?
Yeah, so there are multiple ways that you can do this, and that's what's unique about these enzymes. They're very good at making very large deletions, and so you can make deletions in the DNA anywhere from a few base pairs to hundreds of thousands of base pairs. And because they're right to that nucleotide, it opens a door for things like, say you wanted to get rid of an entire exon or an entire region of a gene, you can do that. And so we're evaluating that for its ability to treat diseases, say, out of hematopoietic stem cells. So can we deliver? The other unique thing about it is that you can deliver it as RNA only. You don't have to add DNA. So the RNA would make the enzyme itself does the work. And it's not dependent on the cell to use the cell's DNA repair machinery. And that's important because we don't have to have that cell going through division. It can be a completely quiescent cell, and these enzymes will still work on that particular cell. So we're evaluating it, and over the past year, really been starting to lay out this pipeline where we would then evaluate and come forward with diseases of hematopoietic stem cell origin, such as some of the hemoglobinopathies. So can we use this as a way to in vivo deliver these RNAs so that we can edit without having to go through cell ablation and things like that.
So what would that in vivo delivery look like? From the patient perspective?
Yes, from the patient perspective, the way that we're setting up that target product profile is that a patient could come in, visit their physician, and it would be an intravenous injection. And so they would come in, an IV injection would go into that, and we're developing and researching the ability to use what are called non-viral things, such as lipid nanoparticles, something where we can take the RNA, encapsulate it in something to protect it, but then have a targeting moiety, so something that would guide it to those hematopoietic stem cells via the blood system. And that's how we're envisioning this product for those diseases.
Yeah, so there's several pieces that you're kind of reinventing along the way. It's not just the first bit that you kind of pitch.
It's not. So you have the recombinases,
yeah,
and that's something that we can take and evolve. And we used a technique called directed molecular evolution. And it's a really big way, but basically what you do is you take the enzyme, you know where you want it to bind, and then you just start screening. And each time you screen, you make different mutations. And so you essentially randomly mutate until you get this enzyme to keep the activity that you want, but now have that activity somewhere else in the genome. So that's the one component. But once we have that, we can make the RNAs, we still need to deliver it. And that's where another set of expertise and research is in how do we take that RNA, put it in something so that we can deliver it with this in vivo. The applicability of the platform is extensive though, because you could do it, we're focusing on in vivo. You could use this for ex vivo as well because of the unique nature of these recombinases and their ability to make these edits very precisely.
How far along are you, and sort of what kind of meetings are you taking here? What kind of partners are you looking for?
Yeah, so I would say internally, the way we're setting up the company, we've got this tremendous amount of expertise coming out of Dresden University. So we have that site in Dresden, Germany, and they're focusing on making those enzymes, doing that evolution to make the enzymes. The science. We've also set up a laboratory and offices in the Boston area, in the US. And what we're doing there, and it's a sort of separation of labor, if you will, in the Boston area, we're really building out the in vivo pharmacology, the disease biology, and then anything that would be required to take something from an idea to the clinic, and then ultimately to commercialization. So what I mean by that is all the IND enabling, the manufacturing, the regulatory, the clinical.
Right.
Is being built out at the Boston office.
And what about from a trials perspective?
From a trials perspective, we're still a little ways away from that. We're focusing now for this year and over the next little bit of time to really build, can we make the enzymes? And we're well along the way on that. Can we deliver the enzymes to where we want? We're well on the way to that. Put the two together and then show that in vivo in an animal model, or that we can actually do this with in vivo. That's one of the things we're focusing on this year. As we get to that, we probably spend the next year, so 2026, now starting to move through that, what we call IND enabling. So anything that we would need, meet with the regulators, get some feedback on what that pharmacology package would look like, as well as what kind of manufacturing processes would we need to have in place to prepare us so that we can move into toxicology studies and ultimately into the clinic.
So being where you are in sort of you've done quite a bit of groundwork, but you're still in the early stages. I'm sure that funding is important and sort of getting how have you found the kind of funding environment, the investment environment in the last few years in the space, and do you feel like it's changing?
Yeah, it's, when you look at companies that are bringing forward a very early novel technology, such as Seamless, there are investors that really are still, as part of their philosophy, investing in the next generation of technology. We've been very fortunate. We have three very good, very supportive investors. Forbion is our main investor. We also have Wellington Partners, and UCB Ventures just more recently came in with us. And so their mantra is really, let's invest in these technologies because that could be the next generation. So that funding for now has gotten us so that we can really put our nose down and generate the data that we need as we start thinking about perhaps a Series A investment to get us into the clinic in a year and a half or something like that from now. So I would say for those kind of companies, it's good. As you get out to the Series B, Series C, I think it's still difficult for genetic medicines companies. And we've been talking to several groups here at JP Morgan conference also around what is this sentiment, what do you need to have as a company, and a lot of the focus and how we're trying to build this as well is taking an awesome technology and then focusing it in an indication where it's completely differentiated. Your technology can deliver an unmet medical need that no one else can do, but also in a disease indication that has a lot of unmet medical need, but also something that can support a commercialization and the funding that would be required in order to go into that.
So those strategic decisions of where you go first, those are really crucial right now.
Very important right now.
Or even the decision to set your operation up in Boston, I'm sure.
Mm-hmm. Yes.
You had a lot of choices.
Yeah.
Why Boston?
There are a couple things into Boston. Actually, some of this was as they were going out and recruiting me. So I joined in February. And it was the board decision, and I think it's a great decision, is as you think about what would be required to really compete in the genetic medicine space, having that in the Boston area, you get access to a tremendous amount of talent, but also there's a lot of funding resource that's available within not only the Boston environment, but the US in general.
Yeah.
And so that's one of the reasons to then start to build that out of the US.
So I know it's day one. I'm sure you've been stuck in this room taking meetings all day, but so far, what are your kind of impressions about the mood at the conference and some of the trends, and what are people talking about? What are people thinking about what this year is going to be like?
I think there's an element of optimism, at least from day one that I've seen. You've just had some announcements this morning about some M&A activity that's starting to pick up.
Right.
And then even more recently, also some announcements of early fundraising and capital raises by companies within the genetic medicine space. So all that collectively has, I think, led to an element of optimism for not only the healthcare in general, but the genetic cellular gene therapy space as well.
Awesome. Well, thanks so much for chatting with me. It's
Yeah.
it's a really interesting space area, and I'll be excited to see how it develops in the years to come.
Yeah, thank you, John.